I am Riley Petersen. I am a graduate student in Sam Butcher's lab at the University of Wisconsin-Madison. And today I'm going to be talking to you about pugs. And no, I am not talking about the little dog with the flat face. I am instead talking about pug or poly-UG RNAs. So these are uracil guanine or guanine uracil, GU or UG repeat dinucleotide sequences. Just a little bit about the nomenclature you'll see throughout this presentation. They can be either orientation, GU or UG, and you can have this little number here on the end, which indicates the number of repeats, 12 in both cases here, or you could have a half repeat, meaning we have 11 complete repeats, and then a single G on the end in this case, an incomplete repeat at the end. But before I really dive in and tell you why I think pugs are so cool and you should be interested in them as well, I first want to kind of take a step back and look at RNAs as a whole. So RNAs have a large level of structural complexity, despite their overall simple sequence composition, right? There's only 4 nucleobases, which if you compare these to proteins where you have, 20-ish amino acids, 4 nucleobases seems really limited. You only have the uracil, cytosine, guanine, and adenine. Yet despite this, you're able to make a large variety of different really interesting structures, as many of you will be very well aware of, such as, riboswitches and pseudoknots, as well as just kind of hairpins and these kind of different double-stranded structures. And this is because of the wide variety of different kind of base pairings that RNAs are able to form. You have the typical kind of Watson-Crick, but you also have things like GU wobbles and Hoogsteen type base pairings, as well as many mismatches that can be formed. But even within these base pairs, there's even another level of structural complexity that is possible with these kind of different sugar puckers, as well as with it being either C3' endo or C2' endo. Although C2' endo typically isn't observed in RNAs, it's more of a DNA sugar pucker. You can also have different glycosidic torsion angles, which is just a fancy way to say the orientation of the base relative to the sugar, being either towards it in a syn orientation or away in an anti-orientation. But you could imagine where if we limit our kind of sequence space even further to, let's say, just hypothetically only including G and U repeats, we could see how this would even further limit the number of possible conformations that these RNAs could hold, where we only really have the possibility to make GU wobble pairs or UU and GG mismatches. So it's hard to imagine what kind of structures these pug RNAs could fold into. And it has been thought in the past that they probably are overall unstructured or maybe only transiently structured. Now we're going to kind of transition as well to talk about repeat sequences as a whole. So pugs aren't the only kind of repeat sequence that are highly abundant within the human transcriptome. Actually, genome. Roughly 3% of the human genome is these dinucleotide repeat sequences. So these kind of simple nucleotide repeats, it could be one repeat that repeats a bunch of time, one nucleotide that repeats a bunch of time, or up to 6 repeats, 6 nucleotides repeating. And these are often referred to as short tandem repeats, short tandem repeat sequences, and they have been shown to play many roles in RNA biology despite their simple composition. They play roles kind of all throughout this mRNA processing, where they can alter maybe where transcription or translation starts, or change how these mRNAs are processed through things like alternative splicing, or maybe they have structures that could affect how mRNAs are processed within the cells and affect their half-lives, how long they can exist, or where the mRNAs end. And of all of these kind of short tandem repeats and all of the roles that they seem to play in cells, the most abundant of these repeat sequences are these GT or CA repeat sequences. And with the GT being enriched within the coding region of our DNA. What this means is that when the DNA is then transcribed into RNA, these GT repeats become GU repeats instead, or pugs. And there's many, many thousands of these sequences within humans. Actually, about 20,000 of them, and they tend to be enriched within regulatory regions of human mRNAs. This means that they have often been linked with many diseases because many of these PUG sequences are actually polymorphic, which means that they vary in length. And as those lengths vary, they are linked with things like cancer or atypical cystic fibrosis, asthma, schizophrenia, heart disease, and other diseases as well. And as interesting as this potential link with human biology is, and I'd be happy to talk to you about it more in the question section or whatever, but we're actually going to be focusing on a totally different organism today, and that is C. elegans. C. elegans are these little transparent roundworms that I'm showing you a picture of here. And C. elegans are really interesting to study as a model organism for a couple reasons. One being that they are transparent. And what this means is you can actually watch the biology happening inside of the living animal as it happens, rather than having to kill it and look at it later. And you can kind of watch the mechanisms as they occur. They also have a lot of similar machinery as what humans have, so they make a really interesting model organism for seeing how maybe this biology could be occurring in humans. But C. elegans have this really interesting property for these pugtails where they're added as tails onto the 3' end of mRNAs being targeted for silencing, which kind of seems a bit more unique for C. elegans biology. And so how this was originally discovered, how it came about to us, was in the discovery of this protein RDE3 by our collaborators here at UW, Marv Wickens and his lab. And RDE3 is a poly-UG polymerase, which means that it adds specifically perfect UG or GU repeats as tails onto these mRNAs. And what our other collaborators at Scott Kennedy's lab at Harvard were able to show is that these pugtails confer silencing activity to these mRNAs. So this is showing the expression of a fluorescent protein called GFP with either no tail or different tails added. And we can see when there's no tail, the GFP glows this really beautiful bright green color. And then as the tail length, as you add these tails, you now have only the pug tail causes this silencing. Now the green is gone, whereas all other tails, you can still see that green color. But it's not quite as simple as just, does it have a tail? What you also have to have is a certain length threshold. So when you have shorter pug repeats, there is no silencing activities. But once that RDE3 moves a little further and we get to a certain length threshold, we now start to see potent silencing activity in C. elegans. And so this is where our lab kind of came in, the Butcher lab. And that is that when we see these kind of stringent length requirements for activity, as a structural biology lab, our brain often goes to, could this be some kind of structure existing? And so we wanted to then go in and test this hypothesis of, is there a structure which is conferring the activity in the living animals? And so the first method we turn to try to investigate this structure is called circular dichroism, or CD spectroscopy. And essentially how this method works is it measures how a sample absorbs circularized light, which will inform you about its secondary structure, as different kinds of secondary structures will have different CD signatures. Here I'm showing an example for proteins. You can see how a beta sheet versus an alpha helix absorbs this light differently, which can then be used to probe what kind of structures exist. And it works very similarly in RNA. And so we did CD on our pugs, and we can see when we have these short repeats where we have no silencing activity in the living animals, we have little to no CD signal indicating that they are more or less unstructured. We then hit that same length threshold where we start to see silencing activity in the animals, and we now have a really strong CD signature indicating a structure has formed. We can complete that repeat and switch the order of a GU or UG, it doesn't matter, it has the same structure. We can also put it in the middle of an RNA by adding A's to the end of it. So it doesn't matter whether it's on its own or in the middle of an RNA. And so awesome, we do have this correlation between some kind of length dependence for silencing and for folding. But we want to know more than just, could it be related to a structure? We want to know what this structure is. And the first kind of hint that appeared to us was actually in the CD signature itself. We look here at this minus 240 peak, this negative 240 peak here, and that's often indicates parallel quadruplexes. So what are quadruplexes, first and foremost? So quadruplexes, or G quadruplexes, as they're typically called, are these four-stranded secondary structures formed by consecutive guanines, or G tracks. So you have this kind of typical G-tracked sequence here where we have a few Gs in a row and then some kind of loop sequence, which could be one or multiple nucleotides, and then more G tracks. These then come together to form G quartets, as I'm showing here, which are these 4 guanines paired together in this cyclic Hoogston type base pairing pattern, where you have 2 hydrogen bonds between each adjacent base. So each base is making 4 hydrogen bonds all together. They're really, really stable, and they're stabilized further by the binding of this metal cation here in the center, this positive metal ion, which has to be either potassium or sodium to help stabilize those structures. They then stack, these quartets stack on top of each other to form these quadruplexes, these stacked G quartets. And the quadruplexes kind of come in 2 major categories. They're either intramolecular or intermolecular, which just describes the number of molecules. So intramolecular means it's one RNA looping back on itself to form these quartets. You can also have an intermolecular one where each strand of those 4-stranded structures, or maybe a couple of different strands, come from different RNAs that are also still brought together. Within this, you can then have 2 different structural classes, either parallel or anti-parallel. So parallel meaning all of the strands are going in the same orientation from start to finish, 5' to 3', or they can be intermolecular, meaning they go opposite directions. I mean, anti-parallel, sorry, meaning they go opposite directions. And so we see this signature in the CD that indicates maybe it's a quadruplex. And so we decided to test a well-known property of quadruplexes, which is this metal cation requirement for it being potassium or sodium. A similar metal cation that won't interfere with other structures necessarily, but will interfere with quadruplexes is lithium. Lithium is known to inhibit or prevent folding of quadruplexes. So we went back to our CD and we made a lithium control, and we can see that now we have, once again, no CD signal indicating the structure unfolds in lithium. So that's another pretty good sign that this is likely a quadruplex. We wanted to go one step further, though, and see. So we used this dye called N-methylmesoporphyrin, or NMM. So it's this kind of porphyrin structure. A porphyrin that maybe you'd be familiar with is hemine, which is the porphyrin that has an iron in the center and is responsible for carrying oxygen in your blood. So it's a very similar structure to that, and it has this kind of interesting property where it binds specifically to quadruplexes, and upon binding, its absorbance spectra redshifts, which means it increases in absorbance. So we can see here when we have unbound NMM, it's at about 378 nanometers for its absorbance. And as we add the RNA to it, we can see that absorbance spectra shifts and increases, indicating that it is binding to the PUG when we add it. And it binds relatively tightly. So once again, this is another line of evidence that this is indeed a quadruplex. But we want to know more than just the category of the structure. We want to actually solve the structure. So now it's time to dive into actually determining the structure of the PUG fold. So the PUGs, we did this using 2 different parallel methods of structure determination, both of which on GU12 RNA, which is the same length that we need for silencing activity. And those 2 methods are nuclear magnetic resonance spectroscopy and X-ray crystallography. So NMR, as it's often called, is a way to measure how different nuclei interact within the molecule. So how the nitrogens, carbons, and hydrogens all are kind of interacting with one another, how close or far apart they are. You can feed all this information then into a machine, which will then help you to build a model to kind of fit those parameters. Here I'm showing you some example of some NMR data where we were able to specifically measure the hydrogen bonds in our G quartets to help build our model. Here I'm also showing you our crystals that we grew for X-ray crystallography. You'll notice how they're kind of this pretty red color, and that's because we utilized NMM, that small molecule I showed you, to help stabilize the structure and help us grow our crystals. So using both of these 2 methods, we were able to determine the structure of our GU12, our PUG fold. So here I'm showing you both of them, and the good news is that they agree. So that's the first step. Each method has its pros and cons. In particular, the number one thing that'll stick out for the crystal structure is that salmon-colored NMM up here on the top, which we had to use to grow the crystals, to help grow the crystals. But there's always a fear when you add a small molecule into a crystal structure that it might be in some way perturbing the structure if it was free. So we didn't have to include the NMM in our solution NMR data. And so we are able to say that the NMM isn't perturbing the structure in the crystal structure. The crystal structure does have a slight advantage, though, of being able to see those potassium ions bound within the center of the quadruplex, where we aren't able to see them in the solution structure. But the good news is all other structural features within these two models more or less agree with one another. And so going forward, I'm going to just focus on the NMR structure, the solution structure, because that's the structure that I actually determined and worked on. But know that everything I tell you about the structure going forward is also true for the crystal structure. So if we kind of rotate that structure and we look at it from the top down, we can now kind of see that signature quadruplex quartets stacking on top of each other that I showed you before. So all of the green G quartets, as you can see here, the G's are all in green, the U's are in blue. So we can kind of see that cyclic nature of the quadruplex. This is an example of an intramolecular quadruplex, so that one RNA wrapping back around on itself. And it's also an example of the parallel geometry. So all the strands start up here at the top, work their way down, and then there's a loop connecting the bottom of the quadruplex back up to the top so it can repeat all over again. And this quadruplex we're calling it an atypical RNA quadruplex, and we're naming it the PugFold. And what kind of makes it atypical? There's a couple major properties. So here I'm showing you Terra, which is a very well-studied example of an RNA quadruplex. And it follows that kind of typical G-tracked, G-rich sequence motif that is typically seen in RNA quadruplexes. But obviously, the PugFold does not. There's no G repeats at all within the sequence. And this actually makes it the first ever example of a quadruplex with no G repeats in a sequence whatsoever. Additionally, it's the first ever example of a left-handed RNA quadruplex, which just describes the overall backbone direct orientation, the direction from 5' to 3', where we can see that it's going kind of counterclockwise for the PugFold, versus in Terra, as well as all other previously determined RNA quadruplexes, they go right-handed or clockwise. So basically, the tail is curling in the opposite direction. So the pug tail is going one way, and all other previously determined quadruplexes go the other. And so kind of zooming out from that top-down view back to the side or working our way down the quadruplex, sorry. We're gonna start at our first quartet. We have the first G quartet. We can kind of see that beautiful hydrogen bonding network here very clearly now. For our first G quartet, we then have this bulged out uridine, which sticks out of the structure. We then move down one more to the second G quartet. We then cap the structure on the bottom with a U quartet. It's previously quadruplexes have been observed that contain mixed quartets like U and C quartets as well as their G quartets. So it's not super uncommon to see a U quartet. And you'll notice that it also has that kind of cyclic fashion that we see with the G quartets, but there's only one hydrogen bond between each adjacent base rather than two. We then also have our final G quartet here, which is connected to the single uridine propeller loop, which also protrudes out of the structure and connects that bottom G quartet back up to the top so the structure can repeat in that parallel fashion. So if we look at all of these kind of together, at once, we can kind of see this very interesting feature of the PugFold where every single position has a different conformation. They have a different combination of those syn versus syn versus anti-glycosidic torsion angles, that orientation of the base to the sugar, as well as those different sugar puckers, and some of them being inverted or upside down relative to the starting backbone orientation. So despite this being a really simple sequence, it has a very high level of structural complexity. And if we now kind of look at just, rather than the top down, we look at the side of a single strand of the quadruplex, which is symmetric and repeats 4 times. So it's the same kind of orientation for each. We can see another couple interesting properties of the quadruplex. The first being this non-sequential quartet stacking order. So we have the G1 stacks on the G3, then the G5, and then the U4 down here at the bottom. So it goes 1, 3, 5, 4, rather than 1, 3, 4, 5. And this is because of multiple backbone inversions which occur. The first at this bulged out uridine here at the U2 position where the backbone inverts. So the G3 and U4 are upside down relative to the G1. The backbone then inverts again at that U4 position, which then allows that G5 to insert itself in between the G3 and the U4, which makes it so that single uridine is able to span just one G quartet rather than having to go all the way from the bottom to the very top of the structure. And with these multiple backbone inversions, as well as that overall left-handed geometry, it made us think of another really interesting RNA structure, which is zRNA. So zRNA is categorized by these multiple backbone inversions, as well as this alternating C2', C3' endo sugar puckers, and this central inverted syn anti-stacking, which is all seen as well in our pug-fold backbone orientations. There's obviously some differences with that bulged out uridine and this kind of flipping down here at the bottom, which doesn't occur in zRNA, but overall the structures are similar enough. We're calling this the first ever Z-form RNA quadruplex. And what this results in is a really highly complex, compact or condensed structure. So we can see here that for the pug fold, the 5' and 3' ends are only 3 angstroms apart from one another. You could practically connect them. Versus if it was a more extended A-form structure, they would be 60 angstroms apart from one another. This really kind of unique, interesting, compact structure could not have been predicted by any current machine learning programs or algorithms for structure prediction. Here I'm showing AlphaFold 3, which has the advantage of being able to add potassium ions as a ligand. And so we can see the structure here that it's predicting for GU12 with potassium ions, and we can see that it's not quite correct. The structure is very different than what the actual structure of the pugfold is. And so this really highlights why it's still really important for some certain level of human supervision for machine learning programs and for structural determinations. We're getting a lot better at predicting some components of RNA structures, but some of them are still a little bit outside of our reach, which is something like Eterna might be really helpful. Eterna, for trying to help develop, humans are very good at pattern recognition, so helping to develop these additional pattern recognition parameters to help us better predict some of these more complicated structures in the future. So hopefully I've kind of shown you and convinced you that this is a really fascinating structure, and it's really, really complicated and nobody would have ever expected it to exist. But we still need to kind of go back and answer the original question that brought us to this structure in the first place. We have the correspondence of that length with both the structure and the silencing activity, but we want to actually prove that the structure is what's causing the silencing activity rather than just kind of the correlation that we're seeing right now. So to test this, we used a couple different methods. The first one we turned to is our good old friend NMM. So we did our previously used injection experiments, where when we inject the RNA with the pug tails, we can see that we have really nice silencing, but then we added NMM to those injections, and now we can see we lose our silencing activity. So NMM is in some way blocking the silencing activity of these pug RNAs, these pug tails. But that still kind of doesn't fully answer the question, because NMM could be blocking the structure from doing its required roles in the silencing pathway, or it could be that the NMM is stabilizing the structure and the structure itself is blocking silencing activity because it needs to actually be unfolded, because NMM does stabilize the structure overall. So we wanted to look at a way to more specifically perturb the structure itself and test how this impacted silencing activity. And so to do this, we utilized RNA modifications, where we replaced all the guanosines in the pug with 7-deazoguanosines, which just removes this nitrogen here and replaces it with a carbon. And what that does is it removes one of the hydrogen bonds in our G quartets, destabilizing them, making the structure fall apart. And we can see that this is true when we look at our CD. We lose our pug fold signature in CD when we have the modification, and the RNA now runs like single-stranded RNA in a gel. And so when we go back to our GFP silencing methods, we can see that we recover our GFP signals when we use the 7-deazo-G modified RNA, meaning that when the pug fold can fold, we have silencing. When it can't fold, we don't. So we're very directly testing that now, and we're able to show 100% that it is the pug fold which is conferring this silencing activity in the living animals. So kind of let's take a step back and talk about how this process works. So what is the pugtail actually doing in this case? So it's working in this process called RNA interference, or RNAi. This is usually triggered by something like double-stranded RNA. These double-stranded RNAs are processed into small interfering RNAs, which bind to an Argonaute protein and use them as guides to find the mRNA being targeted for silencing. These mRNAs are processed, cleaved, and then RDE3, our poly-UG polymerase, comes in and adds pugtails to the mRNAs being processed for silencing. Once those pugtails reach that necessary length, they're now able to fold into the pugfold. This pugfold then recruits an RNA-dependent RNA polymerase, which creates more secondary small interfering RNAs using that RNA as a template, which then feeds back up into this cycle and allows an amplification of this silencing signal. It also allows the silencing to be passed on to the progeny for up to 6 generations. So it causes this process called transepigenetic inheritance. So kind of in summary of what I've talked to you about today, the pugfold is this really fascinating structure, which I've introduced you to today. It's the first ever Z-form left-handed RNA quadruplex, and is the first quadruplex with no consecutive guanosines in the sequence. It's also a highly complex and compact structure that could not have been predicted with any current machine learning structure prediction approaches. It is essential for gene silencing in C. elegans, making it the first ever known example of a functional quadruplex within a living animal. And with that, I'm going to go ahead and thank the people who helped contribute to this project, in particular my PI, Sam Butcher, and our previous postdoc, Christian Escobar, who worked with me on the solution structure, and the staff at NMR-FAM and our collaborators. And with that, I thank you so much for your time and patience and attention, and I'd be happy to take any questions.